Hello guys, welcome to Bosons Botany. So in this video, I will teach about charge calculation in amino acid. So before watching this video, it is very important that you watch the previous video on amino acid and structures, because without having basic idea about amino acid and their structure, you cannot understand this video. I have already discussed about the types of amino acid, their structures, and also triple letter or three letter and single letter code. So I will be using all these concepts in this video, so it's very important you go to that video and then come back to this one. So I will add the link of that video in the comment section, so you can click on the video link and watch that. Okay, so let's start. So before calculating amino acid charge, it is important to understand few concepts. First, what is a zwitter ion? So as you can see, this is an amino acid and this is the alpha carbon. This is the carboxylic acid or carboxylic group which is attached to this alpha carbon, and therefore it is known as alpha carboxylic group. Similarly, this is ammonia group which is attached to this carbon alpha or alpha carbon, so it is also known as alpha amino group. And this amino group is having one positive charge over here, so the net charge of this compound is +1. Now, when this carboxylic acid releases one of its hydrogen ion, then this compound will become a zwitter ion having one negative charge and one positive charge. This negative charge is on the carboxylate ion and this positive charge is on the ammonium ion, giving a net negative charge, and this is known as the zwitter ion. When the ammonium group releases its hydrogen ion, then it will become NH2. And the net charge on this compound is now -1. As you can see, there is only one negative charge here. So zwitter ion is very important because it is having one minus and one plus, or one positive and one negative charge, giving overall zero charge on the compound. So an amino acid cannot remain uncharged. It means it has to have one positive, one negative, or both positive negative charge, becoming a zwitter ion, but it cannot remain uncharged or having no charge at all. As I have shown here, this structure of amino acid is absolutely wrong because it is having no charge. So this structure is wrong. Okay, so just remember this. And zwitter ion is non-polar in nature. Why? Because it is having net negative charge. Although it is having one positive and one negative charge, still the net charge is 0. So zwitter ion is non-polar and therefore it will dissolve in a non-polar medium. So these 2 points are very important. Now, what is pKa, or understanding the concept of pKa values? So pKa is known as minus log of Ka. And what is Ka? Ka is nothing but it is the dissociation constant. So pKa is antilog of Ka value, which is the dissociation constant. Now you need to remember some of the pKa values as I have mentioned here, because these values will help you to calculate the charge of a polypeptide chain. So these are the groups that I have mentioned, and these are some pKa values. Now, before mentioning the values, let me again get back to this structure here. This is the alpha carboxylic group, as I have already told you. This is the alpha ammonia group or ammonium ion group. This is alpha carbon, and this is the R group, right? This alpha carboxylic group will have pKa1 or pK1. This will have pKa2 or pK2, and this R group will have pK3 or pKr values. Okay, so alpha COOH will have pKa1 or pK1, alpha NH3 plus will have pKa2 or pK2, and R group will have pK3 or pKr. I hope it is clear. It is just nothing but nomenclature to differentiate various groups and their pKa values. So let's begin. First we have alpha COOH. Its pKa value is 2 to 2.35. We will take 2 as the value during calculation, and this is the pK1. Now we have two very important acidic amino acids, which are aspartic acid and glutamic acid. As you can see, I have written the 3-letter code, single letter code, and the full name here. So aspartic acid will have pK3 value as 3.65. Why pK3? Because this is the pKa value for the R group. pK3 value for the R group, and R group in aspartic acid and glutamic acid is carboxylic acid, but it is not alpha carboxylic acid. Okay, it is the carboxylic acid which is denoting the R group. So if you do not watch the previous video, then you cannot relate this video. So it's very important you watch the first video on amino acid. So the pKa value for aspartic acid is 3.65, which is the pK3. Glutamic acid has pK3 value as 4.25. Now from here, that is from histidine onwards, we will have basic amino acid and their pKa values. Histidine has pKa value as 6, or the pK3 value for histidine is 6, which is very close to the neutral pH, that is 7, and therefore histidine is used as a buffer. Then alpha amino group will have pK2 value as 9.6. Then cysteine will have pK3 value as 8.18. Tyrosine will have pK3 value as 10. Lysine will have pK3 value as 11, and arginine will have pK3 value as 12.48. So remember, arginine has highest pKa value. It is very important. Arginine has the highest pKa value. So if you understand the trend over here, pKa values for all the amino acids are less, as you can see. But as we go towards the basic amino acids, pKa value will increase. I hope this is clear. You need to remember these values. Now to give you a better understanding, on how to plot pKa values on a titration curve, then this figure is the best example. This is the titration graph for glycine. As you know, glycine is non-polar and its R group is hydrogen, normal hydrogen atom. So it is non-polar and it will not have its third pKa value. So it is a diprotic amino acid. I will be defining diprotic and triprotic amino acids. So glycine is a diprotic amino acid which is non-polar because its R group is having hydrogen ion. It is non-polar. So it will have only two pK values, pKa1 and pKa2. This is for alpha carboxylic acid and this is for alpha ammonia group. So pKa1 here it is 2.3, approximately 2, and amino group will have pK2 value as 9.6, approximately 9. And this is the Pi value. What is Pi value? It is the point on the titration graph where the pH value equals pKa value. Here pKa and pH value will match, right? So I hope it is clear. There are only two pK values in case of glycine because it is non-polar and diprotic amino acid. On the other hand, if we take the example of glutamate or glutamic acid, it is a polar amino acid and it is having 3 pK values. Therefore, it is triprotic amino acid. Let me write polar amino acid and triprotic. So it will have 3 pK values. As you can see, this is pK1 for alpha COOH. This is pK2 for alpha NH3 plus, and this is pKr or pK3 value for COOH group. It is not alpha COOH, it is the COH group for the R group, which is variable in different amino acids. So there are 3 pK values, as you can see, and this is the Pi. This is the Pi, sorry, not this one, this, sorry. This is the Pi value. As you can see, this is the Pi value, where pH equals pKa. I hope this is clear. So, if we categorize amino acids, it is very clearly mentioned over here. Amino acids are of two types, diprotic and triprotic. Diprotic amino acids are all non-polar in nature and they have two pK values, pK1 and pK2. And triprotic amino acids have three pK values, pK1, pK2, pK3, and they are polar amino acids. And to calculate the Pi value in case of diprotic amino acid, we will use this formula, which is pK1 plus pK2 divided by 2, that is taking the average of two pK values to calculate Pi value for diprotic amino acid. And in case of triprotic amino acid, the Pi value is calculated as pK1 plus pK3 divided by 2 for acidic amino acid such as glutamic acid, aspartic acid. And for basic amino acid, Pi value is pK2 plus pK3. So let me write here, this is pK1 for alpha COOH, pK3 for acidic amino acid is COOH. In case of basic amino acid, pK2 is alpha NH3 plus, and pK3 for basic amino acid is NH3 plus. I hope this is clear. Now, as I have already mentioned before, Pi value is the isoelectric point where pH equals pKa value and the net charge is 0. And this definition is very important. Now, amino acids undergo protonation and deprotonation under various pH values. So, let's have a look here. Protonation, when pH or Pi value is less than pKa value. Again, protonation will happen when pH value or Pi value is lesser than the pKa value of that amino acid. For deprotonation, pH or Pi value has to be greater than pKa value of the amino acid, and amino acid undergo protonation and deprotonation. So protonated value or charge during protonation on an acidic amino acid will be 0, and when acidic amino acid undergoes deprotonation, it will have a negative charge. For basic amino acid, the protonation charge will be positive and deprotonation charge will be 0. It's better you understand the concept, but do not memorize these points, because it is only and only cleared after you understand the concept. So I have taken few points here to give you clarity on this concept. So let's have a look. I have taken cysteine, which is having 3 pK values. 3 pK values. And it is a triprotic amino acid. So this is cysteine. As you can see, this is cysteine. This is alpha COH. This is alpha NH3 plus, and this is the R group. Okay. Now, when the pH is 3, what will happen? Okay, before explaining this point, let me tell you the concept of pH first. Okay, I hope you all know what is pH. pH is minus log of H plus ion concentration or hydronium ion concentration. And when the pH is high, that is more than 7 on the pH scale, then hydrogen ion concentration will be less, because it is antilog of H plus ion concentration, so the pH will be more when hydrogen ion concentration is less. And when pH is less, that is less than 7 on the pH scale, then the hydrogen ion concentration will be more. Now let's take this example over here. Suppose this is a beaker number 1. It is having this structure or this amino acid, and we have pH 3. The medium pH is, the pH of this medium is 3. So since the pH is less, so there are many hydrogen ions in the medium, as I have already drawn several hydronium ions. Now what will happen? Under low pH, this NH2 group has a higher tendency to take up one hydrogen ion, as I have shown here. This NH2 group will take this hydrogen ion. Sorry. Okay, so this NH2 group will take the hydrogen ion, and what will happen? This will become NH3 plus. Very simple. Now I have raised the pH from pH 3 to pH 5. Now there will be lesser concentration of hydrogen ion in the medium. Now, to balance the pH or the imbalance in hydrogen ion concentration in the medium, this carboxylic acid will give its hydrogen ion in the medium. First, what happened? When the pH was less, there were several hydrogen ions in the medium, and as a result, NH2 will acquire one of the protons from the medium to become NH3 plus. And after raising the pH to 5, hydrogen ion concentration will decrease in the medium, and to balance this decrease of hydrogen ion concentration, this COOH will release its hydrogen ion in the medium, and as a result we will get carboxylate ion because it has donated its H plus in the medium. And now I have raised the pH to 8, from 5 to 8. Now even lesser amount of hydrogen ions are there in the medium. Now what will happen? This ammonium ion will release its hydrogen ion in the medium, and this will become NH2 again, right? So the concentration of hydrogen ion will be maintained in the medium. So this is the concept of pH and protonation, deprotonation. Here protonation is taking place. This is protonation. This is protonated form. Protonated. This is also protonated form because it is having hydrogen ion, and this is having NH3 plus. So one extra hydrogen ion. And this is deprotonated form. This is protonated form, and they both are deprotonated here. So this is how pH concept works. Now let me get back to this point here. So cysteine, when it is kept under pH 3, what will happen? This alpha carboxylic acid will be protonated, this alpha ammonium group will be protonated, and this R group will be protonated. Now after increasing the pH, as you can see it is pH 5 now. After increasing the pH, first the carboxylic group will donate its H plus. This H plus, this H plus, this will be released, and we will get carboxylate ion having one negative charge. So alpha carboxylic acid is now alpha carboxylate, which is the deprotonated form. Then alpha NH3 plus is protonated, and RHSH2 is protonated. Again we increase the pH. Now it is 9. What will happen? This alpha NH3 plus will give its hydrogen ion in the medium. So alpha carboxylate ion is deprotonated, alpha NH2 is deprotonated, and RSH is protonated. Again we increase the pH. We increase the pH, and now this R group, pH, let's mention here pH equals to 10. Now this R group will release its hydrogen ion in the medium, and now all these three groups are deprotonated. So one thing is, 2 points, not one but 2 points are very significantly important here. First, alpha COOH donates H plus before alpha NH3 plus in the medium, and R group will give last. R group will donate its H plus last in the medium, and increasing the pH, deprotonation takes place. So these 2 points are very crucial, and to calculate the Pi value for cysteine, we will take Pk1 and Pk2 divided by 2, right? I hope this is clear. Okay, now how to calculate charge on a polypeptide chain. Now this is a question which came in CSIR net JRF. So calculate the charge on this polypeptide chain at pH 1. It is mentioned over here, pH 1. Very simple. So this is the polypeptide chain. This is the penta polypeptide chain. As you can see, we have glutamic acid, we have methionine, arginine, threonine and glycine. So before solving the problem, few steps are very crucial. First, you need to write the N terminal and the C terminal. This is the N terminal and this is the C terminal. And N terminal means this has alpha NH3 plus group, and C terminal means this has alpha COOH group. I hope this is clear. And I have written single letter code of these amino acids. This is aspartic acid, this is methionine, this is arginine, this is threonine, this is glycine and this is cysteine. Now what I have done, I have written the pKa values. I have written the pKa values for all these groups. So alpha NH3 plus will have pKa 2 as 9. Aspartic acid has pKa 3 as 3. pK value for methionine is 0 because methionine is non-polar. Arginine has pK3 value as 12. Threonine has pK value 0 because it is non-polar. Glycine has pK value as 0, again non-polar. And alpha COOH will have pK 1 value as 2. If you are getting confused, then it's better you go back to that table that I have explained, because from there I have mentioned these values. So it's very important you memorize those values. Now I have told you that when the pH is less and the pKa value is more, for an acidic amino acid, protonation will happen, and amino acid acidic amino acid, when it is protonated, it will have 0 charge. So aspartic acid, it is having 0 charge because the pKa value is more than pH, or we can say pH is lesser than pKa value. And non-polar amino acid will have 0 charge. Arginine, it is basic in nature, so pH is lesser than pKa value, so it will be protonated and it will acquire 1 positive charge. Threonine, it is non-polar, so it will have 0 charge. Glycine, again 0. And alpha COOH, it is acidic, and when it is protonated, it will have 0 charge. I hope this is clear. Again, I am explaining you. Aspartic acid, it is acidic amino acid, and when the pH is less than pKa value, so it will have protonated form, and the charge on it will be 0. Methionine, it is non-polar, charge is 0. Arginine is basic amino acid, and pH is lesser than pK value, so it will be protonated and have one positive charge. Threonine is non-polar, having zero charge. Glycine, zero charge. Alpha carboxylic group, it is acidic, and pH is lesser than pK value, so it is having zero charge. And this alpha ammonia group will have positive charge. Why? Because it is basic in nature and pH is less than pK value, so it will be protonated having one positive charge. So how many positive charge we have? We have two. So the net charge on this penta polypeptide chain is +2 on pH 1. So I hope this is clear. So thank you for watching, and if you have any doubt, please mention in the comment section. I would definitely solve your queries. Thank you for watching.